Hello, everyone. Welcome to today's webinar. So my name is Han Lu, Tech Support Scientist at ACD. And so before we get started, just a few points. Everyone in the audience will be automatically muted. If you would like to ask a question, please do feel free to select the chat button and type in your questions. If we're not able to address your question during today's section, we will reach out to you and follow up offline. Okay, so in today's webinar, I will be covering the following topics in the order presented below. So first, I will give a brief introduction and overview to RNAscope technology. So what is RNAscope technology? How does it work? What does RNA assay workflow look like? And what is the right assay for me? Next, I'll cover how to plan for RNAscope assay. So there are three main steps involved. The first step of RNAscope assay is sample preparation. So we'll touch upon how to best prepare tissues for RNAscope technology. And then the second step is RNAscope pretreatment steps. This is really the key step for obtaining optimal RNAscope signal. And the third step, the RNAscope assay workflow itself, is actually quite straightforward. It's basically a kit, and most reagents are ready to use, and it's very user-friendly. So in the third part of today's webinar, I will share with you some tips we have for the manual assays and for troubleshooting. I'll give some pointers to individual assays and explain how you can achieve optimal results through the steps of sample qualification and assay optimization. So after that, I will give a brief introduction and to show you the capabilities of RNAscope automation assays. Last but not least, we will go over some frequent asked questions, including one of the most asked questions of an RNAscope application, the dual, how to perform dual RNAscope ISH with IHC or immunofluorescence. Okay, so what is RNAscope technology? It's a novel in situ hybridization technology. On a very high level, the workflow is very similar to IHC or traditional ISH. The assay starts with permeabilized tissue to allow the target probe penetrate into the cell and bind to the target sequence. The specific sequence then recognized by the RNAscope probe will then get amplified through an amplification cascade. The signal can then be visualized under the microscope. So unlike the traditional ISH where a dig or radio label ribo probe is used to detect target sequence, this technology uses short oligo probes. What is then unique about RNAscope? It's really its probe design. So in IHC and traditional ISH, high signal to noise ratio is the #1 desirable feature. And so the unique probe design of RNAscope technology allows signal amplification and background suppression at the same time. So the result of this is a highly sensitive assay that's capable of achieve a single molecule detection at a single cell level. And this assay is also highly universal. So it virtually works for any genome, any gene, in any tissue. So how then is this simultaneous signal amplification and background suppression achieved? The key aspect of the technology is really its unique probe design called a double-Z probe design. So RNAscope probes are short, oligo-based. So each oligo for each Z contains 3 elements. The lower region of the Z is an 18 to 25 base pair sequence that's complementary to the target RNA sequence. The upper region is a 14-base tail sequence, and there's a spacer region that links the upper and lower region of the Z. So the lower region of the double Z, 2 oligos, make up a target-specific binding site that's about 50 base pair. The upper region of the double Z forms the pre-amplifier binding site, and this is where the amplification cascade occurs. So the feature of the double Z design is that it only works in a pairwise manner. So the only way you get signal amplification is when two oligos, two Zs, are bound right next to each other on the target transcript. The pre-amplifier will only bind if two Zs are together, and followed by amplifier binding and then label probe binding. You get signal amplification. And so if there is non-specific binding of any one individual Z oligo to a non-target sequence, there will be no amplification. So essentially, there's no likelihood of having two oligos right next to each other in a completely random manner. And this is how we eliminate the background and achieve signal specificity. So this is a really a highly specific and robust signal amplification system for mRNA transcript detection. So with a standard probe design, each target probe is actually a pool of 20 ZZ oligo pairs that binds to about 1 kb region of target region. So with this double Z probe design scheme, our probe will be able to bind to fragmented RNAs and detect degraded RNAs. Okay, so going back to so the RNA assay workflow and what does it look like. So going back to this slide, again, this is on a very high level, very similar to IHC. RNAscope assay start with permeabilized, properly prepared, and fixed tissue with RNAscope pretreatment reagent to allow the target probe to penetrate into the cell, bind to the target sequence, and the specific sequence are recognized by the double Z pair, then get amplified through the amplification cascade. The signal can then be visualized under the scope as punctate dots. So each dot represents a single copy of RNA. The signal can then be quantified on a single cell level by either manual counting or automated image analysis using software. Okay, so which assay is right for me? There are 4 different types of manual assays to choose from. The 2 main categories are the chromogenic assays and the fluorescent assays. So the chromogenic assays are either singleplex or can also duplex. The fluorescent assays can singleplex, duplex, or do multiplexing for up to 4 colors to detect 4 different targets. So if you need to multiplex more than 2 targets, if you're doing co-localization studies, or if you're going to perform immunofluorescence with antibodies, or your tissue have endogenous GFP or RFP markers, then the obvious choice is the fluorescent assay. The chromogenic assays includes chromogenic brown, chromogenic red, or the duplex assay. They're based on either the HRP, horseradish peroxide, or the AT, alkaline phosphatase chemistry. The red and brown assays are very similar. So the brown, the HRP-based assay, and the red is the AT-based assay. The brown assay is very robust, very sensitive. The stain is permanent, and it is mostly widely used in RNAscope assay. The red color really, stands out and provides bright color stains with high contrast to background. So it's useful for studies with tissues with endogenous color background, such as melanins in skin or lungs from smokers. As you notice, there are different probe channels. There are different RNAscope probe channels. So these are not to be confused with the fluorescent wavelength channels. This is really built for duplexing or multiplexing capability. So a different amplification tree is built for each probe channel. For singleplex assay, the C1 probes are used, and then C1 and C2 probes are used for duplex, and then C1, C2, and C3 channel probes are used for triplexing. So I just want to mention quickly that if you work at a core facility or if you're running high throughput assays, automated RNAscope assays are also available on the Leica BoundRx and the Ventana platforms with the brown and the red assay. So with the Leica platform, it's also capable for performing duplex and multiplex assays. Okay. So let's take a closer look at the amplification schematics. So the amplification cascade starts with pre-amplifier binding to the tail sequence of the double Z probe pair, followed by the amplifier and label probe binding. So the label probe is conjugated with either HRP or the AP enzyme. So by reacting with a DAB substrate or a brown or a fast red substrate, a brown or red color is developed. So a few points to note. So we don't recommend interchanging reagents between the red and brown assay or mixing reagents from different kits or similar assays. This is because all of detection kits are QC tested on the kit level before they're released for optimal performance. And also, as you can see, for the singleplex assay, the C1 probe are used. So C1 probes are provided as ready to use and no dilution is required. So next, let's look at the duplex assay. So the duplex assay is used for detecting two targets. So in this case, both C1 and C2 probes are used. The amplification cascade for C1 and C2 occurs simultaneously. So the amplification cascades I often think of as building the amplification tree. So in this case, two different trees are being built at the same time for the C1 and C2 probe. Specifically for the duplex assay, the C1 probe is developed with a green chromogen and will always be green. And the C2 probe is developed with the fast red chromogen and will always be red. This assay, as mentioned, can also be run on the Leica Bound RX machine, and then can be either red-brown or red-green. So basically, by default, the C1 probe is provided as 1x ready to use, and the C2 probe is provided as 50x. So for the hybridization step, you make a mixture of the C1 and C2 probes and apply on your sample. You can do so by dilute the C2 probe 1:50 with the C1 probe. It's also possible to use the C2 probe alone. In that case, you just dilute the C2 1:50 with the probe diluent. Okay. So what about triplexing with multiplex fluorescence assay? Now, 3 separate amplification trees are being built at the same time. The label probes in this case are now tagged with 3 different fluorophores, Alexa 488, Atto 550, and Atto 647, which can be detected in different fluorescent channels. Again, similar to the duplex assay, the C1 probes are provided as ready to use, and C2 and C3 are provided as 50x. So far, we have reviewed some basics of RNAscope technology. Let's turn to the actual execution of the assays. I'm going to focus on the manual assays. So the first step of RNAscope assay is sample preparation, which includes fixation, processing, embedding, sectioning, and storage. And so it depends on your sample type. We have detailed guidelines for each sample type, FFPE, fixed frozen, fresh frozen, and culture cells. And there's a separate protocol for each sample type. All the information should be included in the user manual part 1. And so make sure you select the appropriate manual. Okay, and then the second step, which is really the key aspect of RNAscope technology, is the pretreatment steps. So I'm going to spend some time talking about the specific recommendations for the pretreatment step. And again, the third step is the RNAscope assay flow. Okay, so as I mentioned, the first step of RNAscope assay is the sample preparation. So shown here is an example of FFPE samples. These are formalin fixed and paraffin embedded. The fixation and storage conditions are, so you fix samples in 10% NDF for 16 to 32 hours at room temperature. The samples are then processed, going through a dehydration series, and then followed by xylene. And the samples are then embedded in paraffin using standard procedures. And the recommended section and size is 5 micron onto, we recommend the SuperFrost Plus slides. And then for storage, we recommend to store sections with desiccants at room temperature for up to 3 months. Okay. So here's an example of the recommended method for fresh frozen samples. So fresh frozen sample is a commonly used sample type for the fluorescence assay. So for fresh frozen samples, a recommendation is to freeze your sample within 5 minutes of tissue harvest in either liquid nitrogen or on dry ice, and then embed the tissue in OCT. And then you can store the block at -80 for up to 3 months in an airtight container. So when you're ready to section, the recommended section size is 10 to 20 microns. And again, we recommend the SuperFrost Plus slide. There's a step here that's actually very critical, is to air dry your slide inside the cryostack at -20 for an hour before you store the sections in the slide box at -80. And that can go for up to 3 months again. So I want to bring to your attention how sample thickness can affect RNAscope signal. For simplicity, this is showing a multiplex fluorescence assay with fresh frozen mouse brain tissue of varying sizes, section sizes. As you can see, sample thickness is directly correlated with signal output using the exact same experimental condition. So therefore, it's really important to follow the sample preparation guidelines, which is the part 1 of the user manual. Okay. So the second step of RNAscope assay is sample pretreatment. So this is to prepare to open up the cells or tissues, similar to permeabilization step of IHC, to allow probe and amplifier access to the tissue. So as I mentioned, pretreatment is really one of the key steps to ensure assay success. So if you don't digest the cell enough, or if you digest too much, in both cases you'll lose signal. Additionally, if you have high background, and you can have high background or poor tissue morphology when you digest the cell too much. So we will visit this topic again during the troubleshooting part of the webinar. And the other point to pay attention is that just like the sample fixation part, we have different pretreat guidelines for different tissue type, FFPE, fresh frozen, fixed frozen, or culture cells. It also depends on if you're running the chromogenic or fluorescent assays. Therefore, it's really important that you have the appropriate user manual for your specific sample type and assay type before starting the assay. So the pretreatment reagents usually involves one or all of the following steps. So H2O2, this is to block endogenous peroxidase, and then target retrieval or epitope retrieval. This is a boiling step for fixed frozen or FFPE tissue that had been fixed for a long period of time. And the step is to remove the cross-linking introduced by the fixative. And the protease digestion step, and this is similar to IHC, to permeabilize the cell and allow probe and amplifier access. So we can look at 2 examples here. So shown here is FFPE samples running the chromogenic assay. And for FFPE, the first step is a baking step and deparaffinize in xylene to remove the paraffin, and then followed by the H2O2 epitope retrieval and protease digestion steps. So once this is achieved, you can comfortably proceed with the rest of the assay. So in this next example, we're showing a pretreat step for a common sample for a fluorescent assay, which is the fresh frozen tissue. As you can see, in this case, the pretreat steps are different, which involves post-fixation, dehydration, and protease digestion. So if your samples are different, please be sure to visit our website for the appropriate manuals for your sample type that we support. In terms of the actual workflow, it's actually quite straightforward. It's probe hybridization, followed by amplification, counter stain, and detection steps. Okay, so for the manual assay, you can run a 1 or 2 day assays. So once you have the slide ready, just make sure to follow the guidelines on the specific protocols for optional starting point. Usually, once you started the protease digestion step, you must proceed with the rest of the assay and then finish the assay. So we just reviewed the 3 main steps of RNAscope assay. And now you have seen the overview of the technology and the workflow. I'd like to share with you some tips for the manual assay for troubleshooting and assay optimization. Okay, so first, be sure to select the, as I mentioned, appropriate user manual for your tissue type and your sample type. And we advise you to follow the protocol exactly as described. Follow the fixation conditions as closely as possible. And then next is to review sample pretreatment conditions for your specific tissue and assay type. And these are both critical for RNAscope assay success. And the third is to use controls with your tissues. And this serves as an assay workflow technique check, and also to confirm RNA quality in your sample before you proceed with the target probes. And then also to review to make sure you have the required materials to ensure success. So here's a checklist of the must-haves for the manual assay. For example, the hydrophobic barrier pen. This is used to make sure the reagents don't leak during the assay. And here, shown here is the help plate or the steamer setup for doing the target retrieval step. So we have protocols for both. It's important to pay attention to time and temperature, as we have found that this is critical for optimal target retrieval. And this is how the RNAscope reagent kit component looks like. So it's 3 parts, and you should have the pretreatment reagents, you should have the RNAscope detection kit, and also the wash buffer. So the probes do precipitate during storage at 4 degrees. So be sure to warm the probes at 40 degrees for 10 minutes before use. Our probes are actually very stable at room temperature of 40 degrees. Also, if you notice any precipitation in the wash buffer, and also take it out and warm the 50x wash buffer at 40 degrees for 20 minutes before you make up your 1x solution. Okay, so the HypEasy oven. So RNAscope assay contains many steps performed at 40 degrees, from the protease to probe to amplification. So the assay is highly sensitive to both temperature and humidity. So the oven is designed to maintain the precise temperature and humidity. So for consistent results, this is a must-have. So you can expect background or reduced signal or both if during any of the incubation steps the temperature is off or there's reagent volume loss. So you can see here the components includes this humidifying tray with humidifying paper, and this is the cover for the tray, and this is the slide rack for holding the slide. And there are 2 options for washing slides, and you can use the tissue tag washing tray, which is very commonly used in the lab, or you can use what we have designed. It's called Easy Batch Slide Processing System, which can help streamline your assay. Okay, so now we have reviewed some overall tips for manual RNAscope assay. I'll give some pointers for specific assays you need to pay attention to. So shown here is the red chromogenic assay. So the tips for the red assay are the mounting media, and make sure to use the EcoMount as a mounting media because a red substrate, fast red, is very sensitive to alcohol. And so do not use any alcohol-based mounting media, and do not dehydrate the sample with alcohol to avoid diffuse signal. And also, it really helps with the diffuse background if you dry the sample in a 60-degree oven for at least 15 minutes before you mount. The other tip for the red assay is the red chromogen, the fast red, is both chromogenic and also fluorescent. So if you're only working with a single-plex assay, but if you want fluorescent, the red kit can be an option versus the multiplex fluorescent kit. Okay, so the tip for the duplex assay is very similar. Again, the probe for C2 is provided at a 50x, so diluted with the C1 probe. Here, the recommended mounting media is vector mount. Again, the red signal is very sensitive to alcohol, so do not dehydrate with alcohol. and then dry the slides before mounting. Okay, so the probe configuration for the duplex assay is C1 will be green and C2 will be red. And so the duplex positive control, provided the PPID is in C1, it will be green. And this is a medium-high expressor. And then polar 2A is a low to medium expressor, and it's provided as a C2 probe, and it will be in red. Okay, so now for the multiplex fluorescence, sorry, one more thing I forgot to mention is that we do recommend to place low expressors in the red channel because the red color really stands out. Okay, now for the multiplex fluorescence assays, there are 3 options for color combination. So there are 4 amplification steps. Steps 1 and 3 are the pre-amplifier and amplifier binding, and M4 is the step where the label probe binds. It depends on which M4 out option that you use, you can put different channel probes in different colors. So as you can see, the C1 probe can be in green or red, orange, and the C2 probes can be in red, green, far red, and C3 probes can be in far red and green. So 2 things to note is that far red is not really visible by eye. And then the green is the least distinct from tissue autofluorescence. So we recommend to put low expressors in red or far red and high expressors in green. Okay. Also, we recommend use the Prelong Gold as a mounting media for this assay. So please visit our website for channel selection guidelines. Okay, so I have given you some pointers to each specific assays. Next, I would like to cover how to optimize the assay and so you can achieve the best results. So how do I get quality data? The first step is really to qualify your sample using controls. So we have semi-quantitative scoring guidelines where we assign a score from 0 to 4, depends on the number of dots you're seeing. So for example, if you have 4 to 9 dots per cell and none or very few dot clusters, we'll assign a score of 2. So the first step is to perform the assay with control slides and with using control probes. Okay, so this is a technique check to make sure the assay flow steps are working. And then, and if your negative control is clean and your positive control PPID is signal as expected, and then you can move on to your own tissue, again using the control probes. So the positive control is used to check the RNA quality in your tissue. For example, we have a QC standard of 4 to 10 dots per cell for the housekeeping gene PPID in most tissues. And the negative control thing will be informative, if the assay flow is optimized for example, if you have over-digested tissue, you can expect to see background signals even in the negative control. And the second step is to get quality data, is to optimize your assays. So, for example, make sure you choose the appropriate fixation condition for your tissue type and follow ACD guidelines. For instance, for showing here for FFPE tissues, we recommend to fix in 10% NBF for 16 to 32 hours at room temperature. So if samples are fixed at, So if samples are fixed at 4 degrees, it's considered underfixed. Also, the 4% PFA is pretty similar to NBS, except PFA is not very stable, especially when it's diluted, and so especially when it's used at room temp. So if you have fixed instead of in NBF in PFA, and then this will be considered underfixed as well. And you can obviously overfix your tissue as well. So the second example is pay attention to special sample type that require different pretreatment. For example, we know in xenografts and organoids, the cells are kind of loosely packed, therefore require a much milder pretreat. And so are lymphoid tissues, such as spleen, tonsil, lymph node, and these tend to require a gentler pretreat. On the other hand, we know liver tissue and then heart tissue, and heart tissue is mostly muscle, and these are rather tough, and so and they might require a stronger pretreatment conditions. Okay, so to summarize, these are the factors that can affect assay performance, as we mentioned already. Fixation conditions as well as pretreatment conditions are both critical for assay success. So for pretreat, we always recommend to start from the standard condition. And oftentimes this will work the best since our scientists have already performed optimizations. Some other commonly seen problems are weak signals due to RNA degradation, also high background due to sample drying out, and hybridization conditions not optimal. So we highly recommend using the barrier pan and the Have-Easy oven. Okay. So I'd like to show here an example of a successful result running the chromogenic red assay. So PPIB, as you can see, the positive control showed overall staining in the tissue, while the target is mostly in the tumor cells, and the DAT-B negative control is clean. Okay. So the third aspect of getting quality data is to perform troubleshooting experiments. I will review some troubleshooting tips for the chromogenic assays first. Okay. So while performing RNAscope assays, you might run into several troubleshooting issues. I will be highlighting some of the more common ones. This is an example of an over-digested sample on the left. I'd like to mention that sample fixation and pretreatment actually go hand in hand. So if you have an under-fixed tissue and you follow a standard pretreatment condition, you're likely going to over-digest the tissue. Some characteristics of over-digested tissue include destroyed morphology, really high background, and you can see very poor hemotoxin stain. So when boiling time in target retrieval is reduced, and you can see that the morphology is restored, and you get really strong hemotoxin stain and no background with the negative control. Okay. So for positive control, PPIB, and this, when the pretreatment condition is optimized, you can get much better signal resolution. The dots are punctate, and also the tissue morphology looks better. Okay, so next we'll look at how to troubleshoot for under-digested samples. Again, under-digestion is likely due to over-fixation. So if you have over-fixed the sample for too long, and then you follow the standard pretreatment protocol, and you get under-digested tissue. So the first sign, of under-digested tissue is really strong hemotoxin, very compact nuclei, and then you can get high background shown here. You can get high background covering the entire nuclei, suggests amplifier trapping. So to address this, we can open up the cell further to allow optimal probe penetration and amplifier binding. So when we do that, this allows results in a distinct punctate dots. This is achieved by extending the target retrieval time to 30 minutes. So another way to address the under-digestion problem is that if you have the flexibility, for example, if you're using mouse samples, it's just to start from preparing fresh tissues and using the recommended fixation time. So as you can see from this slide, in this experiment, you can compare samples being fixed for 3 weeks and versus samples fixed for 24 hours as recommended. You can see the signal is much, much weaker when your sample is over-fixed. Okay, so in summary, we recommend to begin with standard conditions, and then you can observe the signal intensity and then background. If you see high background, destroy morphology, and then, or if you see weak signal in a high background. And then you can adjust the pretreatment conditions accordingly. Okay. So we also, so here's another common problem is seen with the assay. So we also have common troubleshooting issues with the assay flow step. So the main causes are using the wrong barrier pen, not using a high-easy oven, or incomplete deparaffinization. So the characteristic of this type of staining is shown here on the left, where you get this kind of a high, really non-specific background signal with a DAD-B negative control probe. And so just be sure, to follow the ACD guidelines for the deparaffinization steps and use a high-easy oven and a hydrophobic barrier pen. Okay. So another common problem, particularly for fixed frozen tissue, is sample detachment. So in general, we recommend using superfrost plot slides to ensure optimal sample adherence. So if you observe tissue detachment, the likely causes are listed here. We have found that adding an additional baking step can often solve this problem. So I will review the additional baking steps incorporated for FFPE and fixed frozen samples, but these apply to all assay types. So for example, if you observe detachment after the hydrogen peroxide or target retrieval step, you can add additional baking step before hydrogen peroxide. And then if you observe tissue coming off from the slides after protease, and you can add a baking step before that step. So both steps are for 30 minutes at 60 degrees in the circulating dry oven. So for sample detachment with fixed frozen tissues, we recommend to air dry slides at -20 for 2 hours after you section it. This really helps with tissue adherence to the slides. And then you can add a post-fixation step in 4% PSA for between 50 minutes to 2 hours. We found that majority of sample detachment actually happens after the target retrieval step. And then so we recommend adding this additional baking step before the protease step. So if you tried all the above steps and you're still observing detachment, you can omit the target retrieval step and just go directly to use the extended protease time to 45 minutes. Also, please contact support, and we can help address the best troubleshooting path for this issue. Okay, so now I will discuss troubleshooting tips for the multiplex fluorescence assay. So when your assay is successful, you should see images like this based on expression level. So polar 2A is green, and PPIB is red, and then UBC is far red, and this is using the M4L8 option. So UBC is so polar 2A is C1 probe. It's a medium-low expressor. PPIB is a C2 probe. It's a medium expressor. And then UBC is a medium to medium-high expressor. It's a C3 probe. For this assay, the majority of customers use fresh frozen samples. And this is showing how protease pretreat can affect RNAscope signal quality. So on the right, upper right-hand side, showing this is a mouse kidney sample where you can see there actually quite a bit of tissue autofluorescence. Nonetheless, when the sample is treated properly, you can see this really distinct punctate red and green dots. On the left is protease digestion is actually performed at 40 degrees, and the tissue is completely over-digested. Not only you don't have any punctate signal, you can see an increase in background. So on the bottom here, shown is a fresh frozen mouse brain tissue. And as you can see, the background is very clean. The signals are very beautiful and punctate. And again, with over-digestion, what you get is this increase in background and decrease in signal. So one other known issue I alluded to with the fluorescent assay is that due to the long fixation time, FFPE samples are prone to inherent autofluorescence, which can, pose challenges for image analysis. So we now have developed a multiplex fluorescent assay version 2 that addresses this issue. So please stay tuned for the upcoming product development webinars. Also, check your microscope settings and make sure any weak signal is not due to imaging techniques. Okay, so I think I forgot to mention for the fluorescent assay, it's important to use the prolonged gold multimedia. Okay, so last but not least, for troubleshooting unknown tissue preparations, particularly these are for clinical samples or archival samples where the sample preparation method is not known. So what we recommend is to always start with the standard condition, and you can optimize your assay from there. Okay, so so far we have reviewed how to get started and troubleshoot with the manual assays. I just want to quickly mention that the RNAscope assays can also be automated on the Leica BoundRx and the Ventana Ultra and XT platforms. So I can show you, so the 2.5 can run on the Ventana Discovery Ultra and XT platform. And this is showing the automation capability of the Ventana platform from tissue to imaging. So here are examples of RNAscope 2.5 via Brown assay in various tissues. And just like the manual assay, you can see the DAB negative control show no background, and the positive control PPID show strong punctate staining. So currently this platform is for the red and the brown chromogenic assays. Okay, so now this is showing the RNAscope 2.5 assays running on the Leica. So it's showing the automation capability of the Leica platform from tissue to imaging. Again, here are examples of the 2.5 LS Brown assay in various tissues. The DAB negative control is clean, and PPIB showing strong punctate staining. So currently, this platform is capable of running the red, brown, duplex, chromogenic, and the fluorescent assays. Okay, so here's an example of RNAscope 2.5 LS Brown assay, LS duplex assay showing GRIN2C in brown and TVALB in red in the mouse brain. And then here's an example of RNAscope 2.5 LS multiplex fluorescent assay using fresh frozen mouse brain. So shown here are the neuronal markers, V group 2 in green, V group 1 in red, and VGAT pseudo-colored in white. Okay, so last but not least, I'd like to cover one of the most frequent asked questions, which is the RNAscope application is to performing DOR ISH and IHC or DOR ISH and immunofluorescent application. So I'll walk you through some of the data and some recommendations for this application. So this is a double ISH and IHC with red-brown, detecting RNAscope in brown here, and then the antibody shown in red. So, sorry, IHC in red. And this is a non-small cell lung carcinoma showing RNAscope signal in red and green, and the immunofluorescence in far red, pseudo-colored in white. So please visit our website under the Science tab for more information about this application. So the success of this double ISH IHC or or IF assay has been really based on the expression levels of the proteins of interest. With high expressing proteins have worked well. Most cell surface markers will be destroyed during the RNAscope protease pre-treat step, and these may not work. On the other hand, if you have an intracellular protein and highly expressed, and they usually work. So here are some general guidelines for running the RNAscope followed by IHC or IF is to only combine working ISH protocol with working IHC or immunofluorescent protocol. So please try the RNAscope assay by itself on your target first. And then always start with RNAscope first, and do not perform IHC and followed by RNAscope. And the other thing is you don't need to perform any additional pretreatment for IHC. The pretreat already performed during the RNAscope protocol will be sufficient. So again, please visit our website for information about this application. Okay. So some other frequent asked questions are, compatibilities with different tissues. Our manual assay can be used with FFPE, fresh frozen, fixed frozen, and culture cells. And the automated assays are primarily supported with FFPE. Please refer to the user selection guide. And then the key difference between the RNAscope technologies and the IHC for FFPE sample is there's no cooling required during target retrieval. In fact, it's important to, once the target retrieval is done, it's important to take it out and directly put it in water at room temperature, and then dehydrate and proceed to protease digestion. And then the most frequent asked question is, why did I not see signal with positive control? It could be due to sample preparation step, or the pretreatment step is not optimal, as you have seen. Or another likely possibility is it might be RNA quality in your sample. Okay, so you can always contact us at support using the following methods. You can email us, call us, or review our resources on the website. The support is available in US, Europe, and Asia Pacific. Okay. Thank you for your attention. With that, I can address some questions from the question. How about BaseScope assay webinar? We will have a webinar on BaseScope assays very soon, so stay tuned. The second question, very commonly asked, should I use DEPC or RNA-free water? The DEPC and RNA-free water is not really necessary for the RNAscope assay. So you can use either millicule or lab-grade distilled water to prepare all your reagents. Okay, so the next question. Why did I not see signal with positive control? So as I mentioned, there's two possibilities. One is to check your sample preparation and pretreatment steps. For example, if you have over-digested the tissue, if you just chew up the cells, you are not likely to see any signal. And if your fixation method is not optimal, for example, if you have if underfix your sample, and then you follow the standard pretreatment, and then it's just too much, and the cells digested too much, and all your signal is lost. And the second possibility is also possible with RNA quality in the sample. Okay, so the next question is, how flexible is it to switch fluorescent dyes between channels in the LS multiplex fluorescent assay? You can, so for this question, you can change the sequence for adding the fluorophores. Okay, and then there's another question on the Ventana. It says, can we perform RNAscope duplex and multiplex assay on the Ventana discovery platforms? Unfortunately, not at this time, but we will keep you updated with any new product development. I think that's all the questions I get. Again, everyone, thank you for attending this webinar. If we're not able to address your specific questions during this time, we will. You can still type in your questions on the chat button, and then we will be sure to follow up with you offline.